Hi everyone, I'm Shafiq, Senior Lecturer from Department of Pediatrics. For this tutorial, I'm gonna give you a brief introduction on flow cytometry, or FACS in short. So what is flow cytometry? If you break down the word flow cytometry into three parts, it basically means flow, fluid, cyto means cells, and metry means measurement. So basically, when we say we are doing flow cytometry, we are actually measuring the cell in a fluid-based situation. So you're gonna watch this video. So flow cytometry is a process of performing measurements on cells or particles that are in liquid suspension. It is a technology that simultaneously measures and analyzes multiple characteristics of single cells or particles as they pass through a beam of light. So with flow cytometry, researchers can perform many studies and clinicians can make diagnoses to help treat patients. For example, clinicians can determine the percentages of CD4 positive T helper cells. This has the application in monitoring progression of HIV/AIDS, for example. And researchers can study how stem cells mature and differentiate into other cell types, for example. So there are various ways and application for flow cytometry analysis. However, most importantly is that the sample must be in suspension. There's many types of samples that can be run in a flow cytometer: cells, chromosome, bacteria, and as well as synthetic beads. This is basically man-made beads that can emit the signal as well. We're not going to go into detail for that one. So we can also use or analyze cells from solid tissue. However, the cells must be disaggregated before the analysis, or else the tissue will clog the flow cytometer. So basically, the typical particle or cell size that are acceptable by a flow cytometer is from 0.2 micrometer to 50 micrometer. This will cover all those bacteria, lymphocytes, as well as monocyte and granulocytes. So three main important measurements that will be considered in a flow cytometric analysis are: first, relative size; second, relative internal complexity; and thirdly, relative fluorescence intensity. So there are various brands and various specs with various prices that we have in the market. The one that we actually plan for the practical class is the second one, FACS Canto 2. However, due to the current circumstances, we couldn't do a hands-on session, but we try to adjust as much as we can in this current climate. So yeah, so this basically, before I go further, this is basically a very expensive machine. It can range from less than 100K and up to like ₦1,000,000. It all depends on the specification of a flow cytometer. So the main subsystems that will be embedded in each of flow cytometer, regardless of the brand, is basically the fluidic subsystem, optics subsystem, and electronic subsystem. So for the fluidic subsystem, basically it was meant to transport the particles in a fluidic stream to the laser beam for interrogation. Interrogation just means that we analyze the cells, basically. So for optic subsystem, it will include excitation lasers, prisms, and lenses. And these three components will en route and focus the laser beams onto the sample in interrogation area, or we call flow cell. So when particle pass through the interrogation area, the beam will be scattered and any fluorescent molecules on the particle will fluoresce. And the next bit in the optic subsystem is that the lenses on the other side will collect the scattered and fluorescent light, and the light will be directed to appropriate detectors. The last bit is electronic subsystem. So it will consist of arrays of detectors which able to convert the detected light into proportional electronic signals. So basically the electronic signals will then be digitized and further processed and sent to the computer. So for data for each cell particle will be stored in the computer, and this data can be plotted in various ways for further downstream analysis. So basically light scatter display is a term we use whenever we want to look at the size or the granularity or the complexity of a cell. So basically forward scatter, or FSC in short, is used to look at the relative size of the cell. And side scatter, or SSC, will identify the complexity or the granularity of the cells. So for example here, you can see lymphocytes. Usually when we look at lymphocyte, monocyte versus neutrophils, lymphocyte is a lot smaller than monocyte and neutrophils. So hence we can see that it's populated down on the bottom left of this picture, and lymphocyte also have less granularity or complexity, so it also sits lower on the SSC scale. So you see the neutrophil, for example, neutrophil and monocyte has about the same size. However, neutrophil is a lot complex or a lot more granular compared to monocyte, hence it will sit higher on the scale of SSC, which is the y-axis here. So the next bit is the fluorescent intensity display. So typically we use dot plot to display fluorescent intensity parameters. So whenever we do a dot plot, we can plot up to 2 parameters, one on each axis. So for example, for this one, this example consists of two samples that has been stained with 2 colors, APC or red signal, or FITC with the green signals. So basically here we put APC in the y-axis and FITC in the x-axis. So you can see the first, so whenever we put in this crosshair light gating, so gating basically means a region, we assign region to denote the cell population. So for this gating strategy, we call quadrant. So has 4 quadrants here: Q1, Q2, Q3, and Q4. You can't really Q3, but you can't really see Q3 and Q4, but it's the bottom 2. So Q1 is basically a group of cells that we denote as y-axis positive, x-axis negative, which translated into FITC negative, APC positive. For Q2 or quadrant 2, so these are the cells that we denote as y-axis positive and x-axis positive, means double positive. So basically here for Q2, it's a FITC positive and APC positive signals. You can see the it's just subsequently for the bottom 2, but just to add on that each color we can assign a conjugate. So for example, if you stain this actually the antibody APC as a CD4 antibody. So it's a CD4 antibody conjugated to APC fluorescent colors. And let's say this FITC we conjugated with CD8, for example, CD8 antibody. So basically, whenever we say Q1, what are the population of Q1? We can say, we can sub the colors with the antibody. So CD4 positive, CD8 negative for Q1. So for Q2, it's a CD4 positive and CD8 positive cells. I hope that makes sense. So I think that's about it. It's a very basic introduction. Of course, there's a lot more ways and complexity in analyzing flow cytometer data. There's also various applications that we can use, which we haven't touched. For example, we can test for cell cycle analysis. We can test for cell apoptosis capacity, and so on. There's a almost limitless application that we can use the flow cytometer for. But this is just the basic use that I've introduced here, which is basically immunophenotyping. Basically, we want to know the characteristic of the cell, whether it has certain antibody or don't have a certain antibody, so that we can identify the cell population of interest. That's all. Thank you.